This video will show you how to perform restriction enzyme digestion on DNA. Restriction digestion is usually performed so that different DNA fragments can be subsequently pieced together like building blocks through ligation. First, prepare an ice box and thaw the required solutions on ice. You will need a 10 times digestion buffer and the DNA samples to be digested. In this example, 3 samples will be digested, and they are the YFP gene, a linearized plasmid vector, and a promoter sequence. You also need the restriction enzymes and sterile deionized water. Next, prepare a clean PCR tube for each DNA sample to be digested. Label the tubes such that you will be able to identify the DNA sample in each tube. These are the reaction tubes in which the restriction digestion will take place. Next, add either sterile water or the DNA sample into each reaction tube. You should add the component that is required in the largest amount first. In this example, 34 microliters of YFP gene is added into a reaction tube. No water will be added to this tube because the rest of the reaction volume will be taken up by the buffer and the restriction enzymes. For the 2 other tubes, water is added first, followed by the DNA sample. The amount of water and DNA to be added will depend on your experimental needs. Remember to use a new pipette tip for different DNA samples. Next, add 4 microliters of 10 times digestion buffer into each reaction tube. With a total reaction volume of 40 microliters, you will have 1 times digestion buffer in the reaction tube in the end. Do use a new pipette tip when you handle different samples. Next, add the required restriction enzymes into each reaction tube. For example, the XBA1 and PST1 enzymes are added to the tube with the YFP gene. The restriction enzymes should be the final component that is added. When all the required components of the restriction digestion are added into the reaction tubes, turn on the PCR machine. Set the protocol as 37 degrees for 60 minutes for digestion to take place, and then 80 degrees for 5 minutes to inactivate the enzymes. Place your tubes into the PCR machine and start the reaction. You may also use a 37-degree water bath and an 80-degree heat block instead of a PCR machine. After the reaction is completed, retrieve your tubes. The digested DNA is now ready for further experiments. If you wish to verify the presence of DNA in the tube, you may perform gel electrophoresis to check if there are bands of the expected sizes.